Hi, everyone. I'm Andrew, and I'm I think, just hop into introducing the first speaker to just get going with things. So André Bastos is going to be speaking first. He's an assistant professor of psychology at Vanderbilt University in Nashville, Tennessee, in the US. And his work takes advantage of sophisticated neuronal recordings, computational modeling, and optogenetics in order to investigate predictions and the neural mechanisms that are behind them. So Dr. Bastos received his PhD at the University of California, Davis, in 2013, where he worked on the thalamocortical communication. And he was also a Fulbright scholar during that time, traveling to Pascal Fries's lab and examining oscillations in cortical feed-forward and feedback signals. He did his postdoc work in Boston with Earl Miller and Nancy Kopel at MIT and Boston University, respectively, before he joined the Vanderbilt University faculty in 2021, I think quite recently. So, Andre, I think you're able to share because you're a co-host.
Yes. Go ahead and do that.
Yeah, I think we're ready to go.
All right, thank you. Thank you for that introduction. Thanks everybody for coming. I recognize a few people in the audience. Renee, Peter, Lars, Flores. Hello everyone. Nice to see you. Hopefully we can get together again soon in person. And since this is a dinner, I think at the very least, we should at least imagine what our favorite food would be for dinner. So for me, pizza and beer, I don't know. There we go. Now it's officially a dinner. So my talk is going to be with a focus on electrophysiology, and in particular, what the non-human primate macaque model can tell us about what I call spectrolaminar features of cortex that are repeating and in some sense canonical across cortex. So first, let's take a quick step back and just ask, well, what are the approaches that we use in neurophysiology? And indeed, some of this has a direct parallel to the fMRI field. The classic approach was to record a bunch of neurons, or maybe one neuron at a time, in a particular Brodmann area, and then pool together a bunch of neurons, or more sophisticated, doing some kind of population decoding. So we would try to understand one area basically at a time, and that was sort of the limiting factor in non-human primates. But nowadays we have several other approaches. First, we can record from several Brodmann areas simultaneously, and we also can record from different cortical layers. And we also have introduced this idea of frequency coding and LFP oscillations as providing a rich space for coding between different tasks. So I probably don't have to mention how important layers are for cortical processing, but I'll just put a very brief anatomical and a cognitive argument. Anatomically, we know that superficial layers in visual cortex feed forward from early to higher level cortex, and deep layers feed back from higher level to low level cortex. And cognitively, one of the first studies to show that this might be important for something like visual attention indeed showed that it was in superficial layers predominantly where signals distinguished between different attentional conditions in that particular task. So I won't go into the details, but just to say that this notion of layers does seem to bridge between anatomy and cognition, which is a clue that it might be very important. Since then, and specifically now with a focus on the non-human primate field, tons of papers have come out showing that if you pay attention to different layers and you record different layers and analyze different layers separately, you see that all sorts of different cognitive variables, working memory, attention, aspects of sensory processing, predictive processing, consciousness and anesthesia, all of these things involve a differential weighting of different layers. Now, I mentioned also the importance of oscillations, and I'm going to combine these two things for a spectrolaminar framework for cortex. And so I don't have time to get into all of the different work on oscillations, which is huge, but I just wanted to bring up one particular study that we just did, showing that if you perform a correlation, simple correlation analysis between spiking activity and LFP power, this is frequency on the x-axis and correlation strength on the y-axis. And we take spikes in LFP from nearby units, not the same units, so we don't have any potential for bleed through or things like that. But in general, the picture emerges that you have this gamma frequency range, about 30 or 40 hertz and above, positively correlating with spiking, and the reverse, negative correlations in the alpha beta range. So this has been sort of suspected for a long time, but we went in there and just confirmed it using these data from visual cortex, parietal cortex, and prefrontal cortex. So we confirmed that this is indeed a canonical feature. In theta, it's interestingly, it's a little bit of a mixture with both positive and negative correlations, but in the alpha, beta, and gamma bands, we see this canonical pattern emerging. So, well, what other canonical patterns are there, and can we combine these two frameworks, the framework of oscillations and the framework of layers? And one early study that did that was a study that was very inspiring to me by Alex Meyer. He was at the NIH at the time, and now he's a colleague of mine here at Vanderbilt. So we're planning to team up at some point on mutually interesting questions. But this is a laminar probe. So this is the sort of technique that we use in the non-human primate. These laminar probes, they have between 16 and 32 contacts up until now. From now moving forward, we now have access to neuropixels and very high density arrays. So we're very excited about going and moving this, even in an even more detailed sense, as was alluded to a little bit in the beginning. But with these somewhat sparse, I guess you could say, arrays, they're sufficient to uncover this pattern in the LFP, which is quite striking. As you move from superficial to deep layers, the sort of predominant frequency that you see in the ongoing LFP moves from gamma, these squiggly lines, this fast activity, to slower activity in the alpha beta range. And then in my postdoc, one of the first things I did was to record with laminar probes, but not in V1, in the frontal cortex, and not in a visual perception task, but in a working memory task. And again, the details of the task I'm not going to get into here, but what I did find is I was able to confirm this basic pattern that was found in V1, but now in frontal cortex. So in red is the gamma power, the relative gamma power across electrodes. This is relative power on the x-axis, and on the y-axis is laminar depth from layer 4 at 0. So gamma indeed peaks in the superficial layers, and alpha beta in the blue peaks in the deep layers. And we found this pattern consistently across all of these areas, and we recorded an additional 3 areas and found 3 frontal cortex areas and found that as well. And we also did some statistics to show that if you leave one area out and compare one area's gamma beta profile to the remaining areas, you can predict the average of the remaining areas from the area that was left out in all cases. So it's a pattern that generalizes there in frontal cortex. And so what do we think is going on here? So this is our current working hypothesis. I have to say that we're not very far in testing this hypothesis, but that's one of the first things that I'm going to be doing in my independent lab. So what we think is happening is we have superficial layers and deep layers. Now the superficial layers, we think, have these inhibitory cells with shorter time constants. And these shorter time constants, when you have recurrence between excitation and inhibition, what that generates is relatively fast activity in the gamma range. On the other hand, if you have slower inhibitory time constants, you get slower oscillations in the alpha beta range. That is one mechanism. Another mechanism for alpha beta in the deep layers is also, as I point out here, their interrelationship and interconnectivity, specifically between deep layers to thalamus, and then thalamus back up to cortex. So that can be another route to instill these deep layers with slower time constants by virtue of the delay across these structures. In contrast, superficial layers, they do not project to the thalamus. So it's only the deep layers that project to the thalamus. So some combination of intrinsic processing with thalamocortical connectivity is what I hypothesize currently that drives these slow rhythms. But what I wanted to touch on in this talk is to generalize the gamma superficial beta deep motif to even other cortical areas. So really to establish whether or not this thing is truly ubiquitous. So what we're going to ask here is, does the gamma beta pattern generalize across temporal, parietal, frontal cortex, individual animals, and different data sets across labs? So to do this, I teamed up with a friend of mine who is a postdoc at the Desimone lab, Bob Desimone's lab at MIT, and I performed the recordings in the Earl Miller lab at MIT. And we recorded from 3 areas in my study with laminar probes that were placed perpendicular to cortex, so an area V4 and an area prefrontal PFC. We also did a parietal area, area 7A. And in the Desimone lab study, they recorded from left and right prefrontal, and then from 3 areas of visual cortex, LIP, MST, and MT. So an example trajectory is shown here. In MST, we were able to get other trajectories where we were more perpendicular to the cortex. So this was somewhat of a bad example there on the anatomy. But so we recorded from these 4 areas. And the question is, does the pattern generalize? We also use distinct tasks. And again, I won't get into the tasks much in today's talk, but I'm glad to talk about how these patterns are modulated, of course, by cognition. And that's ultimately the question that we want to address. So first, does what we call the laminar power, how do we do this laminar power spectral analysis? So we take ongoing local field potential data and we calculate relative power as a function of channel and frequency. So across each frequency, we normalize the power across channels by the channel that had the maximum power at that frequency. So instead of having absolute power, we go to relative power, which ranges between 0 and 1. And then we can plot this very nicely as a map. So we have frequency on the x-axis, and then the color corresponds to which channel had the most or the least power at that frequency range. So if there was no difference across power between layers, then everything would be sort of a teal color and not really very much. But in contrast, this is one example from this data set. We see that in the gamma range, you get more power in superficial layers, in the alpha beta range that flips to deep layers, and then in the slowest rhythms, delta and theta, there's an additional peak in superficial layers. So we extracted this. We also did basically we summarized the gamma to beta cross location by taking the relative power across a frequency range of gamma and a frequency range of beta and seeing where do they cross. And then we analyzed whether or not that crossover point is something that could be used to consistently characterize areas across this data set. So we looked to see if there was a gamma beta crossover across these different areas. So let's focus, for example, on area MT in the first monkey that we recorded. These are 1, 2, 3, 4 individual probe examples. You have frequency in all of these plots on the x-axis and laminar depth on the y-axis. And for these 4 examples, we do find what we call this characteristic swoosh shape present in these 4 recordings. They're also present in the other monkey. And then if you just look and scan across this, there seems to be a huge amount of consistency with a few exceptions here and there. When you take the average across all of these areas, this is the average of area MT from monkey 1 average of area MT in monkey 2, MST, LIP, PFC, you see that this swish pattern that we had originally seen in V1 and then in a few frontal areas, it really generalizes across these areas. And this is from one lab's data set. When we looked at another lab's data set, the data set that I collected, here we have 2 additional, 3 and 4. Yep.
Okay.
What was that?
Sorry, I think, yeah, it's muted now, so we're good.
Okay. Just so I know, when should I start wrapping up?
You still have a little bit of time, I would say, within the next 10 minutes.
Okay, great. So to continue then, in these 2 additional monkeys, the monkeys I recorded, again, looking across different parts of the brain, V4 in visual cortex, 7A in parietal cortex, PFC, obviously, in frontal and prefrontal cortex, we see this pattern emerging there as well. So we also did the sort of typical thing that people do, which is current source density analysis. And current source density analysis requires a visual stimulus as an input that gives very good excitation of that area. And when you perform current source density analysis, what you're doing is taking the second spatial derivative of the LFP, and where you see an initial sync other studies have shown that that actually maps onto the position of cortical layer 4. So we also see cortical layer 4 in all of these areas, and it maps on roughly to the location of the gamma to beta crossover point that I showed you just before. So to summarize all of this, what we seem to be seeing is this gamma and theta superficial, alpha beta deep, and then in between the layer 4 sync. But these CSD plots, they're very noisy on an individual probe session. You really have to squint to look at them, and they have to be done manually. There's no automated method to do it. Whereas with these plots, they're so reproducible across probes, they don't require sensory stimulation, and they can be, for the most part, automated to actually detect these patterns. So we think that this pattern is much more robust. But we developed a little statistical framework to actually try to see if that was the case. And it was, we used an algorithm called structural image similarity, which is an algorithm that was developed in the field of image processing in order to detect how similar 2 images are to each other. So if you would pass image 1 and image 2 of these 2 foxes into this algorithm, despite the fact that there's 12 small differences, this algorithm would return a value of something like 0.9. The algorithm judges the similarity of 2 images between 0 and 1. So these 2 images would be very similar. If you throw in 2 very noisy images, then you would get a value much closer to 0. And so what we set out to do then is to take all of our probes, to throw them into one big bucket, and then to randomly select 2 folds of data. So fold 1 is in teal, and that produces this sub-average of the blue teal probes. And then sub-average 2 is in red, and that produces this sub-average. And then across all of this probe and one of our animals, and now one of the animals' names has been revealed to you. But anyway, in shrimps, one of his areas, all of his probes, the average of all of the probes looks like this. So then we can create several different folds, in every fold taking half of the data in order to create these sub-averages, which we then will compare. And what this allows us to do is compare the consistency both within area, across monkeys, and across studies. So with this single kind of statistical approach, we can do all of those different comparisons. We can also do the same thing, not only with the power, but with the current source density, which we also estimate on a probe-to-probe basis. As I mentioned, there's considerable more variability in the current source density analysis. So again, we're interested in how consistent are these things. And then to judge how consistent this is, we use a non-parametric statistical approach where we scramble the laminar labels of a particular probe. And then we use this scramble, we perform a bunch of randomizations to create a reference distribution to then say, when we do image similarity between this original image and this scrambled image, how much image similarity is there just in noise, basically. And what that gives us is a reference distribution, which is shown here across a bunch of randomizations. And the empirical value is how similar are two sub-averages, basically, to one another that I just showed. And in many cases, we see that this empirical value beats the reference distribution, meaning that there is indeed a lot of similarity between the images. But interestingly, in the reference distribution, it was actually, in some cases, shifted away from 0. And so what this meant is that the randomized label data was also having, a non-zero image similarity. And the reason for that is that parts of the image contain, for example, 0 CSD, or a relative power that's not changing very much. And so when you have that and you label something that's not, you randomize something that's not changing very much, that results basically in the noise being very similar to the original image. So in any case, we corrected for that by just subtracting the mean of the reference distribution from the empirical value to get a residual value, a meaningful value of this image similarity value. And what that's being shown here. So these, so this residual image similarity of, for example, monkey 1's PFC with monkey 1's PFC. Again, so comparing the sub-averages of the areas, and monkey 2's PFC with monkey 2's PFC. There's not a lot of even internal consistency within the area. Whereas with the laminar power method, in PFC, there's some, it doesn't turn out to be significant, but in all other area comparisons, except one other, or two others, you see that there is significant similarity. So for example, this star right here would mean that monkey 1's MST is predictive of monkey 1's MST when you do this comparison. And even across area comparisons are significant. So area MST can predict area LIP, for example. And in many other combinations, you get generalization across areas. And this is generalization across areas, but within monkey. So always comparing, for example, MST of monkey 1 to PFC of monkey 1. We also then did the cross monkey comparison. So now this is, does monkey 1 predict, does monkey 1's residual, power spectrum of PFC predict or look like monkey 2's PFC? And the answer is yes. Does monkey 1's MST predict or look like monkey 2's MST? And the answer again was yes. And for all of the diagonal area to area across monkey comparisons, there was significant generalization. And then on the off diagonals, it's monkey 1 versus monkey 2 on the upper right, and monkey 2 versus monkey 1 on the lower left. Most of these comparisons turn out to be significant, but that's not the case for the established method of current source density analysis. And we're not saying that this method is completely unusable. Indeed, we do find this initial sink that I pointed out, but it's just that the sort of temporal laminar evolution of that is not something that generalizes across areas. So there's basically just one feature of these maps which are consistent, but there's many features across the spectral and laminar dimensions that generalize in these maps over here. So that's what this analysis is saying. So I'll just wrap up with a very quick discussion. Basically, what we've seen is that these patterns here of gamma and superficial if you just look across the gamma band here in red, gamma in superficial and theta in superficial and beta in deep, this is a pattern that recurs across cortex, across different monkeys, across different studies and areas. What we next want to do is to go in there and actually try to now detect meaningful sublaminar patterns between layer 2 and 3, for example, 5 and 6, and also, of course, look at what features of these different layers at an anatomical level are predictive of these spectral features that we see. And there's a few advantages to using what we call FLIP, which is basically frequency-based layer, our frequency-based layer identification procedure. So FLIP has more consistency compared to CSD, and really critically, it doesn't require sensory stimulation. So we see the same patterns in the pre-stimulus condition compared to the stimulus condition or the active cognition parts of the task. And of course, cognition modulates all of these layers and rhythms, but it's basically what you can imagine is a canvas, that this is the canvas on which cognition works. So you see gamma, beta, theta being modulated, but it's being modulated against this kind of overriding canvas, which is for the most part always present. So that's how I sort of think about it. This method or this observation has extremely high signal to noise ratio, so it only takes a few seconds of data before you can really confirm this pattern, and it's robust to angular, non-perpendicular probe orientations into the cortex, which is an important kind of just practical concern for folks that are doing this type of measurement in animals where, you know, the cortex may not be completely smooth and accessible. So we can go in multiple angles and still get these patterns. So we want to, in future work, continue to explore other cortical areas. We do believe, given our current work, that it's a ubiquitous pattern, but with more data, we'll be able to confirm if it's truly common to all of cortex. And of course, there's other laminated structures that are non-cortical, like hippocampus. We also want to explore different species, including, of course, human. How to do that is maybe a discussion point that we can talk about. And of course, relate these things to their underlying cognitive and computational functions. And so with that, there's a bunch of other anatomy that I wanted to touch on, but we will wrap up, I think, because I think my time is done, just with acknowledging all the people that contributed to the work, and in particular, my collaborator who did the MIT, the other data set, Diego Mendoza Halliday. And thank you all for your attention.
Thank you very much for that really interesting and intensive talk. I think we have one hopefully very quick question from Jeff. And in the meantime, what we might have Flores do is start setting up from there.
Should I stop sharing my screen?
Yeah, that would be great.
Hi, can you hear me okay?
Hi, yes, hello.
Hi. So I wanted to follow up on something that you talked about near the end, which was sort of a invariance across stimuli and tasks. And I guess that surprises me a little bit. So if you do a manipulation which is purported to be very gamma specific, so I'm thinking of like contrast in V1, or a manipulation that's very alpha specific, like an attentional manipulation. If you basically do manipulations which historically have been associated with hitting one power band versus the other, do you still expect this invariance?
Yes, actually. So okay. It's interesting, right? What you just said, that contrast is a very gamma specific finding in V1, and alpha is very attentional specific. So those things are true. But what's also true is that the cognitive aspect sometimes does not explain as much variability in our ongoing oscillation as we might think. So for example, the amount of gamma in V1 may only, maybe 50 to 60% of the variance of that is actually determined by contrast. And for the amount of power variability in alpha may only be, 20 to 40% explained by attention. So that begs the question, well, what about the other remaining variability? What's going on with that other remaining variability? And that's what we're tapping into. We're tapping into that variability that is sort of not task modulated, which is always there. So that stuff is there. And at the same time, those rhythms are getting manipulated by sensory processing and cognitive processing. So it's like both things are true. And I really appreciate the question, because I think, it's something that we should probably quantify, and we should probably kind of really tease out how much of this map is static and how much of this map is dynamic, because there's both, of course, in there. Otherwise, I wouldn't be talking about it, right? If it wasn't interesting for cognition.
Yeah, no, so that's very interesting. So just to be clear, you're 20 and 50%. That's sort of guessing, right? You haven't.
Yes, that's guessing. Yeah.
Okay.
But I would be shocked if I was too far off.
Okay.
Having done these studies myself for a few years.
Okay. Thank you.
Yeah. Thanks.